Usually when we talk about a medical diagnosis, there's this expectation of absolute precision.
Right. Yeah.
It feels like engineering almost. You break your arm, the x-ray shows that jagged white line on the screen, and the doctor just points at it and says, there it is.
Yeah, it's binary. It's either broken or it's not.
Exactly. It's clean, and honestly, it's comforting. We like our problems to be visible and categorized.
But then you step into the world of drug discovery, and suddenly that X-ray machine is just, well, completely useless.
Right.
You are looking at a molecular landscape that is incredibly murky. I mean, for the last century, pharmaceutical development has basically been high-end, highly educated trial and error.
Which is just wild to think about, honestly.
It's the absolute definition of diagnostic muddy waters. You're throwing these incredibly complex molecules at even more complex biological systems, human bodies, basically, and just hoping something sticks to the disease without, causing catastrophic side effects.
And that murkiness is exactly why it traditionally takes over 10 years and billions of dollars just to get one single new drug through trials.
Yeah, easily billions.
If you've ever waited on a breakthrough treatment for a loved one, you know that absolute heartbreak of hearing that a promising drug failed in phase 3 trials after 8 years of hype.
Oh, it's devastating.
But welcome to the deep dive, because today we are exploring the absolute frontier of modern medicine, where that heartbreak might actually become a thing of the past.
We're looking at a profound structural transition in science here. It's the move toward what researchers are calling pharmaceutical superintelligence, or PSI.
PSI.
Yeah. This is a world where AI doesn't just predict how biology might react, it actually actively invents brand new functional molecules from scratch to force a specific reaction.
And the sources we're diving into today, for you listening, are this massive stack of technical white papers and architectural blueprints. They detail a complete autonomous AI drug discovery pipeline.
7 massive steps.
Exactly, 7 steps. And our mission for this deep dive is to map out how this prompt-to-drug pipeline actually works. And then answer one really crucial question. How does a radical new training method, something called polymath learning, turbocharge all these specialized AI tools so they don't just, hallucinate nonsense?
Which is the biggest hurdle, really. Moving from stochastic lucky guesses to a deterministic reality requires a completely new way of thinking about how AI agents reason.
Okay, let's unpack this.
Yeah.
Because before we can invent a miracle cure, we need a central brain to coordinate the effort. I mean, how do you even control an AI system this massive without it just breaking down into chaos?
Well, you don't build one giant AI. You build a system of systems. That's the orchestration layer of the PSI framework.
Okay.
You have highly specialized cohorts of agents. Some act as chemists, some act as biologists, others are clinical planners. But the true breakthrough is how they communicate.
Right, the protocols.
Exactly. They use advanced communication protocols, things like Google's A2A or Anthropic's Model Context Protocol, or MCP. And those prevent what we call walled gardens.
Walled gardens, meaning the chemists and the biologists usually don't speak the same language.
Historically, yes. In human labs, an insight from the manufacturing team about how a drug crystallizes might take months to reach the early-stage chemists.
Oh, wow.
Yeah, it's disconnected. But here, these protocols act like a universal translator at the UN. The orchestration layer ensures that a chemistry agent can instantly share memory and context with a biology agent.
Okay, a universal translator makes sense, but let me push back for a second here.
Sure.
Even if the science agents are talking perfectly, science isn't usually what stalls a drug. It's the paperwork. It's the FDA compliance, the regulatory hurdles. Does the AI just hand a human a massive pile of data and say, here, go file the paperwork?
Not anymore, actually. That orchestration layer includes regulatory automation. There's a tool called AutoIND.
AutoIND, yeah.
And it uses Retrieval Augmented Generation RG to draft the investigational new drug dossiers automatically.
That's crazy.
It takes a process that usually requires 100 hours of human labor and shrinks it down to about 3 or 4 hours.
And the regulators just accept that, like an AI writing its own compliance report?
They do, because the system is designed to provide regulatory-grade evidence. There's another tool, ToxMCP. Every time the AI makes a decision, say, ruling out a molecule for liver toxicity, ToxMCP forces it to compile auditable risk questions.
So it shows its math.
Exactly. It complies with LCOA plus principles. It leaves a perfectly legible trail of its reasoning that human regulators can actually verify.
All right, so our AI translator has the team talking and the paperwork filed. But before we build a drug, we need a target. We need to know what disease mechanism we're actually trying to stop.
Yes, the biological target.
Right. And failures at this stage, aiming at the wrong target, contribute heavily to that 90% clinical failure rate we talked about.
Because if you aim at the wrong biological mechanism, it really doesn't matter how the perfect your drug is. It just won't cure the patient.
So how does the AI fix that?
The suite uses tools like TID Pro Target Identification Pro. It uses machine learning on OLDOC and text metrics to predict if a target will actually succeed clinically. And then BioReason Pro steps in.
And BioReason Pro bridges the gap between raw genetic data and actual human disease, right?
Yes, exactly. It deduces a protein's exact molecular function using things like interpro IDs and gene ontology biological processes.
The sources mention something here called finding hotspot tokens using modules like AllSites. What does a hotspot token actually mean?
Think of a disease-causing protein as a complex three-dimensional machine. The AI doesn't just point at the machine and say, break this.
Right.
It maps the precise structural interfaces to find the exact molecular exhaust port, the hotspot that a drug needs to plug into to shut the machine down. Oh, and external tools like Apex can even prioritize antimicrobial targets specifically.
So the orchestration layer is like our AI CEO, Tidy Pro, is the scout finding the enemy base, and BioReason Pro is the sniper pointing out the exact exhaust port to hit.
That's a great way to put it.
But knowing where to shoot is only half the battle. How do we build the actual missile?
Which brings us to generative design. Now, the AI has to generate a high-fidelity molecule from scratch that perfectly fits that exhaust port.
And it uses something called Proteina complexa, right?
Yes, Proteina complexa. It unifies generative modeling with test-time compute, and it's trained on this massive synthetic dataset called Teddy Karmer.
Synthetic datasets. Why synthetic?
Because experimental, real-world data on how proteins perfectly bind together is actually really rare and expensive to produce. So researchers took existing protein structures, digitally chopped them up, and forced the AI to learn millions of synthetic biophysical interactions.
So it basically gives the AI an intuition for how atoms want to physically fit together.
Exactly.
Okay, here's where it gets really interesting to me. The blueprints highlight a tool called ISO-DE, the Isomorphic Labs drug design engine.
Ah, yes, ISO-DDE. What's fascinating here is how it models out-of-distribution events.
Yeah, because it finds what they call cryptic pockets. And when I read the description of this, it honestly sounded like pure magic. Explain how a pocket on a protein can be hidden.
It's pure biophysics, but it does feel like magic. A cryptic pocket is a hidden active site on a protein that simply does not exist when you scan the protein in its natural resting state.
Wait, how can it not exist? Is the protein physically shifting shape like a transformer when a drug gets near it?
That is exactly what's happening. Proteins aren't static statues. They are dynamic, moving molecules. A cryptic pocket only physically opens up and becomes detectable when a very specific molecule approaches and induces a conformational change.
That is wild.
Traditional drug scanners completely miss them because they only look at the resting state. But ISO-DE can model those complex movements over time. And then tools like InuOmni, RosettaFold, AllAtom, and Bind step in for simultaneous design.
Meaning they design the drug and the pocket at the same time.
Basically, yeah. They embed active sites into novel folds so the molecule fits perfectly.
And as it's doing this, the AI is constantly refining the molecule, right?
Yeah.
Using these digital cleanup crews.
Yeah, refinement models. Things like Interformer, Carbonara, and FlowPacker. They optimize the structures by in-painting missing side chains and fixing non-physical artifacts.
Like what the sources call steric clashes.
Exactly. A steric clash is when two atoms in a digital simulation are accidentally occupying the same physical space. It's a glitch. These tools scrub the design to ensure the molecule actually obeys the laws of physics.
Which is a perfect logical transition here. Obeying the laws of physics in a computer simulation is great, but a digital molecule is completely useless if it turns into a puddle of sludge at room temperature.
Right.
Or if you can't actually mass produce it in a stainless steel mat in a factory.
Industrial scale-up is the ultimate reality check. You can design the most brilliant molecule in the world, but if it requires, negative 200 degrees to remain stable, it's never becoming a pill.
So how does the AI solve the factory problem before we even synthesize the chemical?
By interfacing with heavy-duty industry simulators, the suite uses the CPRO agent system. It utilizes something called the parameter cohort to optimize manufacturing variables, things like reactor temperature and residence time.
Okay.
And there's a 4-stage pre-screening process to check for thermodynamic stability. But the real star here is May-C off. It uses machine-learned interatomic potentials
Okay, break that down for me. What is an interatomic potential?
to accurately predict how a physical chemical will behave. Say how the ligand will physically pack into a solid crystal so it can be put in a pill capsule. You normally need quantum mechanics.
Right.
But running quantum mechanical simulations for complex drugs takes an absurd amount of computing power and
It could
time.
take years, probably.
Oh, easily. But by using Macy-Off machine learning, the AI can deliver the extreme accuracy of quantum mechanics at the computational speed of classical force fields.
Wow.
Yeah, it predicts exactly how the drug will crystallize in the real world in a fraction of the time.
That is just incredible. We can design a molecule, find a hidden pocket that only appears when it shapeshifts, and figure out how to crystallize it in a factory all inside a simulation. But
There's a but.
there is. A vat in a factory isn't a human body.
No, it is not.
What happens when this perfect digital drug actually enters a patient? Because if the X-ray is muddy waters, the human metabolism is a complete black hole.
This is the great filter of drug discovery: pharmacology, ADMET, and toxicology. ADMET stands for absorption, distribution, metabolism, excretion, and toxicity.
80 met gap.
Right. This is where most drugs die, in what the industry calls the efficacy trough.
Hmm.
The human body is a highly efficient machine designed by millions of years of evolution to destroy and clear out foreign chemicals.
So the AI has to accurately simulate how the liver is going to attack the drug.
Exactly. The AI basically acts like a virtual human liver. First, you have the UniPK framework ensuring thermodynamic consistency. And then you have these end-to-end metabolite predictors.
Like biotransformer.
Yes. Biotransformer 3.1.0, Deep Metab, Sigma. What they
Yes.
actually do is breathtaking. They don't just predict if the drug will clear the body. They literally generate the exact chemical structures of the metabolites.
Wait, let me make sure I understand this. The AI looks at the drug, predicts exactly where the human liver's enzymes will chop the molecule in half, and then creates a 3D model of the new pieces.
Yes. Tools like GLM SUP and Deep 450 identify the exact sites of metabolism. And then PreCMB estimates the relative changes in those metabolite abundances. It predicts exactly what the human liver will turn your drug into.
But wait, if the liver chops our carefully designed drug into new pieces, we now have completely new or untested chemicals floating around in the patient's bloodstream.
Exactly.
What if those new pieces are highly toxic?
That is precisely why step 6 in the pipeline is quantitative systems toxicology, or QST. Predicting the breakdown is useless if you don't test the byproduct.
Okay, so how does it test them?
It uses platforms like DELISM, vToxinet, and Toxicol. And they take those newly generated metabolite structures and run them through causal perturbation models, tools like Medea and Excel.
The sources mentioned Excel is a 4.9 billion parameter model. Causal perturbation meaning it simulates how a human cell actually reacts when perturbed or disturbed by the chemical.
Yes. It's not just basic pattern recognition like looking at a list of known poisons. It uses biologically constrained insights to predict things like hepatotoxicity, liver damage,
Right.
or cardiotoxicity.
Right.
It simulates real-world intracellular concentrations to see if those new chopped-up pieces will accidentally trigger a cell death sequence.
Okay, so we know it won't poison the liver. But human biology is chaotic. I mean, I'm a healthy adult, but what about a patient with kidney disease? Or a 5-year-old child? You can't just guess the dosage.
And that's where Foundation PBPK transformers and in silico twins come in. Before a real clinical trial ever begins, the AI synthesizes high-fidelity virtual patient populations.
Virtual twins.
Yeah. Tools like InsideRx account for patient genetics, organ impairment, dose, and exposure duration. We can precisely simulate the outcome for a pediatric patient entirely in code to flatten that phase 2 and 3 efficacy trough.
So what does this all mean? We have this brilliant digital assembly line. We're translating between chemists and biologists, finding cryptic pockets, predicting quantum crystal packing, running virtual livers, and testing virtual twins. It sounds flawless, but there is a massive glaring bottleneck in the blueprints.
Always is.
All of these specialized AI tools, the virtual liver, the toxicity scanner, they need data to learn. And high-quality experimental biological data is incredibly scarce. We do not have a billion failed real-world drugs to train this AI on.
Right.
So how is the AI actually getting so smart across all these different fields without massive amounts of data?
This is the pivot point of the entire architecture. This is why we are talking today. To make pharmaceutical superintelligence work without needing millions of costly real-world failures, researchers had to invent a radical new training method.
Polymath learning.
Yes. Polymath learning. Extreme data efficiency. The study calls it one sample to rule them all.
Because historically, to teach an AI a complex skill, you fed it 10,000 curated flashcards, right?
Right. But researchers discovered that if you use highly optimized multidisciplinary samples, you can trigger massive cross-domain reasoning from just a handful of examples.
But they don't just pick normal data. The sources talk about the Lima score, the less is more score. They specifically seek out data with a low Lima score, around a 6. They actively look for weird data.
Right. A high Lima score means the data is typical. It's highly representative of standard, boring patterns. If you train an AI on that, it just memorizes the average.
Okay.
By selecting an outlier, a weird, intensely complex problem, you force the AI to stop memorizing and start learning fundamental, abstract principles of problem solving.
And if they can't find the perfect weird sample in nature, they build it. They created what they call a synthetic prime.
The synthetic prime is absolutely fascinating. It's an engineered training prompt that forces the AI to wear multiple hats simultaneously.
How does that work?
Well, it starts with a biology component, say an 11-base paired DNA sequence. Then it adds chemistry detailing the exact hydrogen bond energies holding those biological bases together.
Okay.
Then it layers on physics, stating the molecule is irradiated with 400 nanometer light and requiring the AI to use Planck's constant.
To calculate the exact number of photons needed to physically break the chemical bonds of the biological DNA, it's like a turducken of science. Physics stuffed inside chemistry, stuffed inside biology.
Exactly. Fusing those domains into one single problem forces the model to connect them through a mathematical lens all at once.
So instead of giving the AI a million simple flashcards to memorize, Polymath learning gives the AI a single, impossibly complex Rubik's Cube. By solving that one cube, it learns the underlying physics of the universe, and suddenly it's a better biologist, a better chemist, and a better toxicologist, all at the same time.
Which answers the crucial question of how this applies to the AI drug tools we just talked about. Drug discovery is the ultimate polymath problem. By training the orchestration layer, or early generative tools like ISO-DE, on these synthetic primes, the AI inherently understands downstream consequences.
Meaning it understands cross-domain causality.
Yes, it learns how solid-state crystal packing, in step 4, inherently affects how the virtual liver will clear the drug in steps 5 and 6. It understands the physical causality between the chemistry and the biology without needing a database of a million examples to prove it.
And from my perspective, looking at the safety of all this, polymath learning triggers the most vital behavior in this entire system. Self-verification.
It organically induces internal reflection.
The AI starts double-checking its own work. When you read the reasoning chains in the transcripts for tools like ToxMCP or CPRA agents, the AI suddenly pauses mid-thought. It literally starts outputting terms like, wait, verify, and let me reevaluate.
Because it understands the interconnected physics, it knows when something doesn't add up.
Yeah.
If a toxicity agent is evaluating liver damage or a manufacturing agent is predicting a reactor temperature, the AI will autonomously pause and say, call a quick Python script to verify the math of its own predicted chemical reaction before it initiates an expensive wet lab experiment.
It acts as its own safety net.
Yeah.
Which completely prevents the AI from hallucinating a physically impossible drug, saving millions of dollars and potentially saving lives.
It replaces blind statistical prediction with rigorous cross-domain deduction.
Okay, let's bring this all together. We have gone on quite a journey today.
We really have.
We started with A2A protocols coordinating the mission, ending the walled gardens between chemists and biologists. We saw Proteina Complexa designing molecules and ISO-DE finding cryptic pockets that shift like transformers.
Yep.
We navigated the virtual human liver, predicting exact molecular chops, and tested the results in silico twins. And we realized that this entire pipeline is powered by the extreme data efficiency of polymath learning solving the ultimate Rubik's Cube.
If we connect this to the bigger picture, the economics of therapeutic discovery have permanently changed. We are replacing sheer data volume with precision data design.
It's a total paradigm shift.
We don't need to fail 90% of the time in the clinic anymore, causing heartbreak for patients, because the AI can synthesize the physics, chemistry, and biology before the drug is ever physically created.
It is breathtaking, truly. It's a shortcut to understanding the next era of human health. And there's one more, but while I was reading through these architectural blueprints, there was one footnote toward the end that actually stopped me in my tracks.
Oh, the DGM.
Yes, the Darwin-Gödel machine, or DGM.
It is the logical, albeit slightly unsettling, next step.
Yeah.
It allows AI agents to autonomously rewrite their own underlying source code to improve their capabilities.
Right. So let's play this out for the listener. If polymath training teaches these models how to reason and self-verify across different scientific disciplines, and the Darwin-Gödel machine allows them to actually alter their own code based on that advanced reasoning, what happens when the AI starts engineering its own polymath synthetic primes?
It's a profound question. We engineered the first synthetic prime using human physics and human chemistry. But what happens when the machine decides our physics just isn't efficient enough?
Exactly. Will the next generation of drug discovery AI invent entirely new branches of physics or chemistry? Will it use concepts and dimensions that human scientists can't even comprehend just to cure a disease?
Once the machine begins generating its own scientific axioms, that X-ray analogy from the beginning completely falls apart.
Oh, totally.
The AI isn't just showing us a clean break on a screen anymore. It's showing us dimensions of biology we didn't even know existed.
The muddy waters might just turn into an entirely new ocean. It is a staggering thought and definitely something for you to ponder as we step into this wild new world of medicine. Thank you so much for joining us on this deep dive. Keep questioning, keep learning, and we'll see you next time.